Assalamu alaikum. It is an immense pleasure for me to start this session as a moderator. My name is Dr. Kashif Asghar. I am research scientist and biobank manager at the Department of Basic Sciences Research, Shaukat Khanum Memorial Cancer Hospital Research Center, Lahore. And my co-moderator is Dr. Naila Malkani. She is an assistant professor at the Department of Zoology, Government College University, Lahore. On the behalf of the organizing committee, we welcome you all to the cancer research session. This session will highlight advances in genetics, immunology, proteomics, and immune therapy. We welcome and thank our speakers who are leading scientists and clinicians. It will be a great opportunity for us to hear from them.
Thank you. So our first talk will be a virtual talk by Dr. Asim Amin. He is a consultant oncologist and currently working at Atrium Health Levine Cancer Institute. He's an associate professor at Wake Forest University, USA, and he has joined us online. So let's begin the talk, please.
Responses, but they're neither requiring any treatment nor are they progressing. Now, what are the agents that have resulted in all of this change in natural history are listed here. Let's see what they are and what is their data briefly. Now, in the 1980s, 1990s, folks who were working with T cells, the effector arm of the immune system that recognizes tumor cells as bad guys and takes them out, it was recognized that these T cells have various switches on their surface, and these switches were called immune checkpoints. So on the left, you will see a whole bunch of on switches that include CD28, ICOS, 41BB, and so forth. And on the right, you'll see a whole bunch of off switches. So the off switches include the PDL1, CTLA4, PD1, and LAG3. And we'll talk about these three because these three modalities actually have been approved. So how do these immune switches work? So when a tumor comes in, it sheds off some of its antigen, and that is shown here as this yellow blob in the center. This yellow blob is taken up by antigen-presenting cells that are specialized cells of the immune system that present that antigen to the T cytotoxic or T killer cells. Once it is presented in the context of self-MHC molecules, that is when the T cell gets charged and gets ready to go and attack the tumor cells. Now, nature always has checks and balances in place. One of these check and balance molecule is the CTLA4 molecule. That stands for cytotoxic T lymphocyte antigen protein 4. And this protein results in putting brakes on the activated T cell. So the T cell initially gets activated and then is dampened down by the CTLA4 molecule. Using an anti-CTLA4 antibody, and ipilimumab is already approved, can result in the T cell now not getting switched off, because remember, CTLA4 was the off switch, so we've broken the off switch, and thereby induces T cell cytotoxicity. Now, some of these activated T cells, when they enter the tumor has protective mechanisms against the cytotoxic T cells as well, and these are in the form of molecules called PDL1 or PDL2, that can interact with another off switch called program death 1 on activated T cells and result in the T cell getting switched off. So the tumor cell is protected from the T killer cell. Using either an anti-PD1 antibody or an anti-PDL1 antibody, the PD1 axis is disrupted. So nivolumab, pembrolizumab, cemiplimab, dostarlimab are already approved for PD1 inhibition, and durvalumab, avelumab, atezolizumab are approved for the PDL1 inhibition. Where do they work in the overall immune response? Here's a tumor cell. The tumor cell sheds off some of its own proteins, the tumor antigens that are taken up by the antigen-presenting cell, presented to the T cell. T cell gets activated and then is suppressed by the CTLA4 molecule. So the anti-CTLA4 antibody blocks this step. This T cell, when gets into the tumors, is inhibited by the PD-L1, PD-L2 proteins through the PD-1 molecule, and that is blocked by the PD-1 inhibitors. So these are the two sites in the immune response that are inhibited and result in greater tumor activity. For that discovery, Jim Allison on the left and Tasuku Honjo on the right received their Nobel Prize in 2018 for CTLA4 and PD-1 inhibition. What was the pivotal trial in melanoma that led to this overall increase in survival? Was the Checkmate 067 study that randomized advanced melanoma patients to either CTLA4 plus PD-1 or PD-1 or CTLA4 inhibition alone. And the overall response rate with the combination of nivolumab plus ipilimumab, that is PD-1 and CTLA4 inhibition, was close to 60%. 23% of these were complete responses. And as I showed you before, those folks who had a response, a whole bunch of them, almost 3/4 of them who received dual immunotherapy did not require any further treatment. This is just to show you what is the extent or durability of these responses. This is a gentleman who was treated for this large melanoma in his cheek. This was not the only site, as you can see out here in the CT scan below. There were several other sites. 3 months later, after 4 doses of combination immunotherapy, he had had a complete response. And the radiologist, in fact, reported that this mass had been resected in the interim. This patient had had no surgery or radiation. This is a neglected melanoma. You can see this large left-sided neck mass that corresponds to this huge mass on the left side. These kind of patients previously would have lived not more than 2 or 3 months. He was treated with the combination of CTLA4 and PD-1 inhibition, and then taken to surgery and radiation. 5 years later, the gentleman still remains in response. But this is clearly a big, huge step in treatment of melanomas that we previously could never think of. The most recent approval of a new immune checkpoint is the anti-LAG3 antibody. You will remember that here's on the left is the tumor cell that is presenting the antigen to the T cell in the context of MHC. At this point, to avoid autoimmune reactions, nature has a checkpoint called LAG3 that inhibits the T cells. And by blocking the LAG3 antigen, the LAG3 protein, the T cell does not get turned off, does not get a negative signal, and remains turned on to kill the tumor cells. This data was presented recently and updated by Georgina Long earlier this year. This is the progression-free survival. Blue curve is a combination of anti-LAG3 plus anti-PD-1, and the green curve is the anti-PD-1 antibody. There was statistically significant improvement in progression-free survival that amounted to 22% improvement and led to the approval of this agent. This is now being tested in the adjuvant setting. Okay, so from immunotherapy, let's switch gears a little bit to targeted therapy. So in 2002, it was reported that 50% of the melanomas have a mutation in a gene called the BRAF. What is this BRAF gene? So here's a cell. All cells have receptors stuck into the surface of their cell membrane. And there are various growth factors that are roaming around that come and they can get lodged into these receptors. This receptor lodging into this growth factor lodging into the receptor constitutes an activation signal that needs to be somehow communicated to the nucleus so the cell can divide. And there are various pathways that can do that. So kind of like a relay race, there is phosphorylation of the tyrosine kinase receptor. That phosphorylation is an activation signal that leads to activation of a bunch of docking proteins and then eventually a protein called RAS. The RAS activates something called RAF. RAF activates something called MEC. MEC activates something called ERK, and then the activated ERK goes into the nucleus and tells the cell to divide. That is what should be happening. Now, what was seen was that the BRAF is mutated in 50% of the melanomas, and that results in this pathway becoming autoactive. So it does not require any upstream signaling. This protein in it by itself is autoactive and keeps on driving the cell to divide again and again. It's a bad thing. Until we found a bunch of small molecules that were developed to block two links in this relay of events, the BRAF inhibitors, vemurafenib, dabrafenib, and encorafenib, and the MEK inhibitors, trametinib, cobimetinib, and binimetinib. Using these targeted agents, you can induce very rapid responses and very significant responses. This is a patient just to show you how rapidly this patient was progressing. The tumor was positive for the BRAF V600E mutation, was initiated on BRAF MEK inhibition, and within 3 months had complete healing of the entire lesion. When this patient was sent to surgery, there was no viable tumor left in that lesion. So what is the overall response rate and outcomes? They're shown here, up to 70, almost 70% response rate with BRAF and MEK inhibitors for folks who have high tumor burden and rapidly progressive symptomatic disease. This is the durability of response with BRAF and MEK inhibitors. At 5 years, 1/3 of these individuals are alive and doing well. You will recall that the 5-year overall survival for immunotherapy is about 50%. One of the problem areas in oncology is always brain metastasis. And what we saw in the Checkmate 204 study was that using nivolumab and ipilimumab, that is CTLA4 and PD-1 inhibition, we can induce responses in about 55% of the folks who have intracranial involvement of melanoma. And 20% of these responses are complete responses. This was completely unheard of until immunotherapy was tested in this setting. This data is still young, but looking at the progression-free survival curves and the overall survival curves, the initial data appears to be quite promising, where there appears to be durability of response as seen out here. This is a patient who had these brain metastases on the right side, was treated with BRAF and MEK inhibition, and these lesions completely resolved within a few months. So both targeted therapy as well as immunotherapy can induce dramatic and significant responses intracranially, and we know that they can be durable. So are we done with melanoma now? No, I don't think we're done because we know that the response rate is about 60 to 70%. So there's 30 to 40% of the people who still don't respond. And so they somehow need to be treated. And we also know that the 5-year survival is about 50%. So we know that half of these individuals will lose their response over time. over 5 years. So, so it is not all done. What we have now found out is that if the tumors, and what you see out here is the gray cells are the tumor cells and the red cells are the T cells. We see that if the tumors are heavily infiltrated with killer T cells, then they respond very well to immunotherapy, as opposed to those cells, those tumors that have a very few T cells, or the T cells are excluded out into the periphery. So the tumors with a lot of T cell infiltrate, they have been termed hot tumors, and the others have been termed cold tumors. And the whole idea with immunotherapy now is to change these cold tumors to hot tumors. And in that, we've seen 3 immune checkpoints being used. There are several other that are now being looked at. So these are this is just a partial list of all the immune checkpoints. The other area of interest is to find out, hey, why is it that some tumors respond and one don't, and some don't, and to actually get tumor biopsies, see if we can change the tumor microenvironment and make it more conducive for the immune system to work. And in that, this little cartoon shows how different strategies to tailor treatment to inflamed versus deserted tumors is being used, using HDAC inhibitors for the excluded tumors, and using intratumoral injection of TLR agonists or sting agonists in this setting. So that's where I think I will end. My conclusions at this point are that there are now effective options for advanced melanoma that was inevitably a fatal disease. We know that almost half of these individuals will get durable responses or cures. Treatment on a clinical trial would still be preferred. That is how we advance the science. And when you're treating a patient, tailored choice of therapy based on patient and tumor characteristics. This alludes to if individuals are healthy and are candidates for dual immunotherapy, they should get that. If the tumor volume is very high and patients are very symptomatic, their tumors harbor the BRAF V600E mutation or K mutation, they could be considered for cytoreduction with BRAF MEK inhibition and then considered for immunotherapy. So work is not clearly finished, but we are far from where we were. I thank you for your attention.
Thank you, Dr. Asim, for an interesting talk. The session is open for discussion. Do you have any questions? Yeah, please. Mike.
I can't hear the question.
So my name is Dr. Ijaz. I want to ask, how does the management of mucosal melanomas differ from the cutaneous melanomas? Do we use the same principles to treat mucosal melanomas, for example, rectal melanomas or GIT anywhere?
Yeah, no, I think that's an excellent question. And that remains an area of very active
Dr. Asim,
research.
can you hear us?
I can hear you. Can you hear me? Are you able to hear me? Are you able to hear me?
Can you please repeat the question?
So my name is Dr. Ejaz. I want to ask that how does the management of mucosal melanomas differ from cutaneous melanomas? Do we use the same principles to treat mucosal melanomas which are used in cutaneous melanomas?
Yeah, are you able to hear me? Are you able to hear me?
Yes.
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 Oh, okay. So, that is an excellent question. So all melanomas are not the same as we know, and as you allude to, they are malignancies of the melanocytes, but the clinical course is very different for cutaneous melanomas versus mucosal melanomas versus the choroidal or uveal melanomas. The mutational distribution is also different. While 50% of the cutaneous melanomas may harbor the BRAF V600E or K mutation, almost none of the uveal melanomas ever have BRAF mutations, and hardly any mucosal melanomas will have BRAF mutations. There is a higher incidence of c-KIT mutations with mucosal melanomas, almost 20 to 25%, as opposed to 1% for cutaneous melanomas and 0 for the uveal melanomas. So the responses for uveal melanomas have not been not been in keeping with the data that we saw earlier. They're not usually BRAF positive. They're usually the response rates to immunotherapy are much poorer. We don't have any specific options. There was some Chinese data presented at one point, but that has not been replicated with temozolomide-based treatment. I don't know. So can you see me now? Okay. So they asked. So yes, and most of the mucosal melanomas have been excluded from the clinical trials that have been done. So that is an area of unmet need. c-KIT inhibition has shown responses in about 25 to 30% of these melanomas, but they're generally not as durable as immunotherapy responses.
Thank you. Is there any other question? Okay.
This is Asim Farooq. I am a research officer. I have a general question about immunotherapy for cancer. How safe is this as compared to, for example, chemotherapy? If we are giving immunotherapy and we are blocking the immunoregulators, so is it not going to cause significant immunopathology in patients?
Yeah. So no, I think that's a very pertinent question, because as we switch over, and I think we will see more and more disease sites adopting immunotherapy and chemotherapy going to the by side, we will need to learn how to effectively manage what are called immune-related adverse events. And these are quite unlike chemotherapy. So you're correct in pointing out this is not chemotherapy. What we're trying to do is we're taking the brakes off these T cells. And what we're trying to do is really walking a fine line between causing autoimmunity and inducing anti-tumor effect. So if you were to imagine a speedometer, and that's how I usually explain that to the patients, if your T cells are normally cruising around at 50 miles an hour and they now see a tumor and they recognize it as non-self and they respond to it through CTLA-4 activation or B, and then the B7 activation, the CD28 activation. Once they're activated, Now we have to make sure that they don't go past a goal threshold. And I kind of arbitrarily set that goal for, just for understanding, is at 135 miles an hour. So from 50 miles an hour, we're trying to go 135 miles an hour. But there is no limit. So there are folks whose immune systems will go 200 miles an hour. And when they start doing that, they will start attacking their own organs. And that is, the most ominous ones, of course, are autoimmune colitis, where the colon can be really inflamed and needs to be managed carefully with immune suppression. We normally use high-dose steroids, and if need be, then go on to higher levels of immune suppression with infliximab. For autoimmune hepatitis, if high-dose steroids are not working, then we may need to go to transplant level immune suppression with agents like mycophenolate, mofetil. And so, meaning it can be any organ system. It can be the pituitary, it can be the thyroid, it can be the adrenal. So, people can develop type 1 diabetes, people can develop autoimmune pneumonitis or autoimmune hepatitis, or any itis really that you can imagine, depending on what speed their immune system is going and what organ system specifically is chosen by their immune cells. So yes, it's tricky. It's not a simple, straightforward cookie cutter thing. There's a lot of nuanced training that needs to go in that.
Thank you, Dr. Asim. From this, we will move to our next talk, which is by Professor Jacek Gonwald. He is a professor of genetics and pathology at Pomeranian Medical University, Poland, and he will present.
Thank you.
And he will present the importance of micronutrient research in the management of oncological patients.
Hello, everybody. Thank you for invitation. I would like to. Okay. I would like to tell some words about microelements. Yes, and why microelements. Yes. So we started our story with, let's say, cancer genetics, and we observed persons with mutations. Family with mutations, and we noticed that penetrance is not 100%. Yeah, in BRCA carriers, only 70, maybe 80% of people are affected. 20% are not, maybe 25 not. With our mutations also not. And of course, there was an extensive study on modifier genes, and they helped a little bit. But we all the time thought that it will be some other factor which influence, and of course about environmental factors we discuss, let's say, from the beginning of history of cancer. So we started to make some kind of registries and just to control and measure some other values, including microelements, and look how it maybe it is association with cancer or not. So my talk will be about, some numbers, yes, what we had. And look, it is cancer risk, probability of detection of cancer, and probability of survival within cancer with some other measurements which can help in prognosis or not. And look at this slide. So we took a blood sample on diagnosis, and after 5 years we observed what happened with this group of patients who were affected with breast cancer. And so there is a selenium concentration. If it is low level, after 5 years we have 33 dead patient and 104 alive. And in opposite side, if selenium is high, so we in a group of 136 patients, because it is divided in 136, all these 4 groups. So in a group of 136 patients, only 15 died and 121 is alive. And if we make statistics, so group 1 versus group 2, 3, and 4. So it is statistically significant. The hazard ratio is 2.5, yes. So of course it is much better to be in group 4 than in group 1. Another, after 10 years, what happened? Yeah, so in group 1 with low selenium level, we have 46 dead versus 88 alive. And if selenium in diagnosis was high, so over 95, so only 16 patients thank you. Only 16 patients died. So of course it is difficult to say at this moment if we control selenium, and for example at the beginning it is low selenium level, and we add, it will help it in treatment. But these numbers indicate that if at diagnosis selenium is low, so probably it is high risk group. Yeah, so maybe treatment should be more aggressive. And of course at the same time, so it will be on the end, we started some trials and try to control selenium level within treatment. So we'll wait for answers. And it is the same breast cancer group, and let's say for measurements of patients to make life, for clinicians to make life, let's say, easier, which selenium, we divided group in, let's say, less effective, so below 70, and that 28 versus 32, and the most, let's say, promising, so this level, 95, 102, not too high. So in this group, only 2 died. Of course, it needs to be verified on long observation. Zinc, yeah, another microelement, the same model. So patients divided in 4 groups with low zinc level and with high zinc level, yeah, and the same here died 43 versus 91 alive. They were in this range. And on opposite side, if it was high, only 22 died and 113 alive. And odds ratio is 2.4, also results statistically significant. So another microelement which can help put the patients in let's say, high risk group of death or low risk group. And the same, below 700 looks bad, and in this range, only 1 patient died in 31. Yeah. So also it needs verification. Selenium level and risk of death with larynx. So again, low selenium level, very high risk of death, 55 died versus 21. And measurement was done 10 years before. And here, 13 are alive and 13 died and 61 are alive. So again, looking at as a factor, risk factor, so patients who can keep selenium on high level, so they are in much better situation. And all this group, it is the same staging and so on. So it is not that this were in a high stage and it is low stage. They were measured according to staging. And the same zinc and blood level, we observe also some differences between serum level and blood level. And in some circumstances, it is better to measure microelements in blood. Yes. And again, there is a group of zinc in blood level with, let's say, we could high risk group of death, 30 died, and 31 are alive, and here only 14 died and 48 are alive. Malignant melanoma, yeah, so again, 10-year observation, selenium level, and we have a model. So here, low selenium level at diagnosis, 78 are alive, 16 died, and here 92 are alive if selenium level is high, and only 2 dead. Also quite significant indicator for survival. And prostate, also we observed in prostate such situation. Again, low selenium level, 32 died, 59 alive. And if it is high, 10 are alive and 83 died, yes. And it is combination, yes. So extreme parts were taken together. And if it is below 70, 35 versus 64. And if it is over 85, 10 died and 87 are alive. Yeah. So it needs, and it is 5-year survival. And again, and zinc. So these microelements, selenium and zinc, looks very promising for further research. Yes. Also, if it is low, so we have this number, 36 died versus 57, and if it is high, so 9 versus 88, and after 5 years. So at the beginning, so we can measure, and then we can predict what will happen in, let's say, 5 years or 10 years. And it is cuprum, and for prostate also it worked. So it is opposite, low cuprum. So it is indicator for good survival, so better survival. So 10 died and 83 are alive, and cuprum, if cuprum is high, 36 died and 57 are alive, and extreme taken groups. So odds ratio is even 10. Yeah. And survival in zinc level, yes. So if zinc is on low levels, so it is indicator for short survival, and if it is on high level, it is promising that survival will be better. So in summary, serum selenium level is strong prognostic marker for survival in cancers. So this which we study and we observe, not in all cancers we observed such association, but in larynx, breast, pancreas, lung, so we observe it, yeah. And pilot study, there are additional prognostic markers and microelements, yes, so we study on this, and there are genotypes improving speciality of selenium levels. So these modifiers here, with combination of microelements, will make more precise prediction of 5-10 year survival. And what about probability of detection of cancer and cancer risk? So it is also difference. What is probability of detection of cancer and cancer risk? So, and just in a second. So again, at diagnosis, we measure some microelements. And so at diagnosis, we in colorectal cancer, we measured selenium. And if selenium was low, yeah, so in this group, we had 55, it is matched cases and controls, healthy controls and cases. 53 cases had a cancer, and 12 were healthy, if selenium was low. If selenium was high, in this group, 25 had cancer and 78 were healthy. So if we make, let's say, measurement, like let's say we make colonoscopy, we make genetic testing and so on, but we also can use this tool just to make some kind of population study. And if somebody is below 46, so maybe it is indication for colonoscopy in this group. Maybe it is, let's say, early cancer. And we did it, yeah. And in patients with familial family history of colon cancer. And if we make, let's say, screening, colonoscopy screening, so we detect one polyp in, let's say, about 20. Yeah. If we make this, and these levels of selenium are low, so we detect about 4 times more frequently polyps in colonoscopy. So of course it is, if our budget is short and we have to, let's say, make some choice who should be tested. So this is, let's say, relatively inexpensive tool that to select patients where we have a chance to detect early cancer or polyps in group of patients. Cuprum also can be used, yeah. So in this group, and colorectal cancer. So if cuprum is low, so we have 11 cancers and 47 healthy. And in opposite, if it is high, so 117 cancers versus 47. So again, much higher probability in patients with higher cuprum level, much higher probability to detect patients with cancer. Yeah. Again, another tool just to help to diagnose patients, or to at least to select for other study. Selenium concentration and lung cancer. Yeah. Again, at diagnosis, we measure selenium. In group below 60, it is 37 cancers and 12 healthy. And in opposites, it is if it is over 80, 14 cancers and 27 healthy. Yeah. So it is quite opposite like here, 37 versus 12 and 14 versus 27. Another, it can be used as an indicator for, let's say, computer tomography. Yeah, who should be selected. And cadmium level and concentration and lung cancer, similar, it also can be used. It is not so strong like before, but also can be used as an indicator. Cadmium on low level, 27 versus 48, and it is 76 versus 52. So serum or blood concentration of selenium, arsenic, cuprum, cadmium are strong markers of colon cancer or lung cancer, and they can be used as a, let's say, make more density in screening, increase the chance to diagnose early cancer or pre-cancer stages. And we did some prospective study. So we had a cohort of 1,700 patients, yes, and we measured some microelements at the beginning, and then we observed 4 years what happened. And within 4 years, we diagnosed 109 cancers, and 56, almost 1,600 stay healthy. Yeah, And in our clinic, there are most of cases of families with breast cancer predisposition. That's why we have for sure in our group overrepresentation of breast cancer, and we observed. So we had 67 breast cancer, 6 ovarian cancer, 5 colon, and so on and so on. So these malignancies were detected within this 109. Yeah. And also we did it within BRCA carriers. So 1,327, and we observed them for 39 months. And in this time, we diagnosed 74 breast cancer, 18 ovaries, cervix, and so on and so on and so on. And we looked what was the level, if here we have arsenic level. If arsenic level was low, so we had 428 patients, yes, with low arsenic level. So in this group with low arsenic level, we diagnosed only 4 cancers. Yeah. If arsenic was high, so in this group with this period of time, we diagnosed 47 cancers. So 2 years, not 2 years, but it was 4 years before, we measured arsenic, and then in group with high arsenic level, we had much more cancers than in group with low arsenic level. So it indicates that it is not only marker of diagnosis, but probably arsenic, if we influence arsenic and keep it on low level, so probably it can be helpful, yes, and can save some, patients, and some cancers will not develop. So still this research is ongoing. And in BRCA carriers, again, also arsenic level, low level, it was 655 and 32 cancers. And if arsenic was in this range, 71 cancers. Yeah. So again, high arsenic level was associated with cancer risk. And selenium, also if it was too low, it was more cancers. In this range, it was the best, but if it was too high, it was again, probability of developing of cancer was higher. And we observed it also, so we made, I think, 15 years ago, such study where we supplemented BRCA carriers with selenium. And after 3 years, we observed that we have more cancers in selenium group than who were not supplemented, who had placebo. And fortunately, we were doing measurements of selenium at the beginning, because at the time, recommendation of World Health Organization, it was to give about 200 micrograms per day of selenium. And for breast cancer, or let's say families with high predisposition to breast cancer, too high selenium level can be toxic. At that time, we didn't know about it. So fortunately, we had measurements, and people who had selenium level, women who had selenium level on low level at the beginning, so they had a profit. Who had high level, so it was toxic for them. Yeah. So at the beginning, we should measure micro elements and then influence. So we have now we know more, and we make some prospective studies. So we have a project, prevention of females malignancies in families with hereditary breast cancer by personalized optimization of selenium level in the organism. So there is a special protocol. We measure selenium at the beginning. Women have information. It can be dietary indications. So there are some microelements they can take. Yeah, so we recommend what is, and we measure selenium level, and according to our experience, the level which is the best for women, yes, with the low probability of developing cancer. So we keep ladies in this group, and of course there is another control group. So, and then we will calculate if we can really influence on cancer risk or not. And there is also another program, and so with BRCA carriers, yes, so they are divided in some groups. There are also controls without mutation, and this extensive study on over 7,000 women, they are going on. So I hope soon, let's say, I think we need some 2 or 3 years, yes, when enough people, when time will be enough long to measure how many people develop cancer in, let's say, recommended group and group just observed group. So then we will be able to answer the question, we can, in this relatively easy way, to decrease number of cancers. It is similar project with arsenic, yeah. And I can conclude that microelements are strong factor in treatment and prevention of cancer. We will see if it is only marker, or maybe we can influence the level and decrease the cancers. It is the group of the most involved people, yeah, in this study. And thank you very much, and welcome for collaboration.
Okay, our third talk is also a virtual talk, and it is by Professor Sarah Zeniven. She is professor at the Institute of Cancer Sciences, University of Glasgow, Scotland. And she will be presenting using the mass spectrometry proteomics to unreveal how to target the tumor microenvironment in cancer.
Some of our recent work, where we have, you will see, extensively used mass spectrometrics to understand the role of the tumor microenvironment, particularly in breast cancer. Now, my group specifically focused the research on triple negative breast cancer and high-grade serous ovarian cancer because they are highly aggressive tumor types, and there are very few therapeutic options against the cancer cells. However, if we look at the composition of these tumors, so here is a staining of an ovarian cancer metastasis in the omentum, you can see how extensive is the stroma in this tumor. So the stroma here is indicated in brown, where we have down staining for anti-alpha smooth actin, which is a typical marker for cancer-associated fibroblasts. So the question that we have is, can we then, instead in this tumor, try to exploit the tumor microenvironment to effectively treat these tumor types? We are particularly interested in cancer-associated fibroblasts as a target, and cancer-associated fibroblasts mostly generate from normal fibroblasts that are resident in the tissue where the tumor develops, and then in the presence of the tumor, they become activated. And what is becoming more and more evident is that now there are different subsets of these of CAFs, which has different functions. And in my lab, at the moment, we are particularly interested in those that are called MICAF, that are those cancer-associated fibroblasts that produce vast amount of extracellular matrix. And we are particularly interested in understanding how these cells can influence the behavior of cancer cells, immune cells, and also endothelial cells. And the reason why we are interested in these CAFs that produce a lot of extracellular matrix is because now if we stain In this particular case, we are going back to an ovarian tumor in the omentum. So this is a metastatic tumor. And we stain the omentum for collagen 1 and 3 that you can see here in red. You can appreciate how little of collagen you have in the normal omentum, but how much you have actually in the omentum in the presence of tumor. So we have really a lot of extracellular matrix that is produced. But why is that important? The reason for that is because it has been known for a very long time that the extracellular matrix can influence many aspects of tumor development and resistance to therapy. So work from my group, but many other groups has, for example, shown that the extracellular matrix can influence the growth and invasive behavior of the cancer cells. My lab has also shown that the matrix itself is able to modify the function of the blood vessels in a way that facilitates the intravasation of the cancer cells to form distant metastases. But it's also known that the extracellular matrix can induce chemoresistance, and there are more and more evidences that the extracellular matrix can also influence the behavior of the immune cells, including the polarization, the activation of the T cells, and also can form a barrier to basically block the entrance of the immune cells in the tumor. So basically blocking, impeding the contact and the killing of the cancer cells. So now the question is, how can we then target the extracellular matrix? And this is a very important question because it's quite a conundrum, the targeting of extracellular matrix, because if you look in the literature, it has been shown that targeting extracellular matrix can either promote tumor progression or strongly inhibit the progression of the tumor and the formation of metastasis. So we really need to find a way to properly target extracellular matrix so that we inhibit their tumor promoting functions. And in my lab, we are particularly interested in targeting the metabolism of the cancer-associated fibroblasts in order to block the production of matrix. And we are particularly focusing on distinct metabolic pathways. So one are those pathways that can actually influence the production of acetyl and methyl groups, because these are very important to epigenetically regulate CAFs. And it's known that some of the functions that the CAFs acquire upon activation are actually under epigenetic control, for example, histone modifications. But also we are interested in amino acid metabolism. And this is thinking, if you think that when the normal fibroblasts get activated into CAFs, they have to make a lot of extracellular matrix, suggesting that they need an additional production of amino acid to be able to support this increase in protein synthesis. And we are vastly using modern mass spectrometry proteomics to basically determine what are the mechanisms that can underpin this production of extracellular matrix. So we typically use shotgun proteomics. We typically start with the complex protein mixture that we trypsinize, digest into small peptides, which can then be fractionated and analyzed at the mass spec to quantitatively analyze proteomes. But we can also, from this peptide mixture, enrich for peptides that contain specific post-translational modifications, such as phosphorylation or protein acetylation, because these, of course, are very important to determine the function of the cells. And then we also analyze this by mass spec and use quantitative approach to accurately quantify them. Now, in terms of models that we use to study CAFs, we mostly work with the patient-derived samples. So we isolate our CAFs from a tumor. And for the story of today, I will focus on breast cancer. So we are isolating CAFs from breast tumor tissues. We also isolate a normal fibroblast from adjacent normal-like breast tissue. And now we immortalize our normal fibroblasts in CAFs, and then we use them for experiment. Now, what you can see here, this is an image where you can see the matrix produced by normal fibroblasts and CAF in culture, and you can appreciate how much matrix the CAFs produce. So they really maintain this ability to produce more extracellular matrix. And if we particularly focus on collagen, that is one of the components of the extracellular matrix, you can see that CAFs derived from the same patient compared to the normal fibroblasts always produce more collagen. So here basically each color corresponds to normal fibroblasts and CAF coming from the same patient. So, but what is in this extracellular matrix? So here we did extensive mass spec proteomic analysis of the matrix produced by CAFs in culture. And what this analysis told us is that we can identify thousands of proteins, but of those only around 300 make most of the extracellular matrix mass. And collagen 6, 12, and 1 are the collagens that are most abundant. And this is important because particularly in breast cancer, these 3 collagen types have been shown to be tumor promoting. But are CAFs also producing similar matrix in vivo? To answer this question, we also performed a proteomic analysis of tumors that grew in mice that were co-transplanted with the patient-derived CAFs and with human breast cancer cells. And what you can see here from our mass spec analysis, we determined that also in these tumors, collagen 6, 12, and 1 are the most abundantly produced. But importantly, we could also show that also the tumor stroma that form from the mouse tissue also contain collagen 12, 6, and 1 as most abundant collagens. The other important information that we got from this analysis is that more than 30% of the CAF-derived extracellular matrix is collagen, and we will see later on why this is important. So, but are these collagens also highly abundant in patient samples? So to answer this question, we use previously published single-cell sequencing data of triple-negative breast cancer. And here you can see, we look at the abundance of these collagens in different subtypes of CAFs. As you remember, we are particularly interested in this MyCAF that produce extracellular matrix. And you can see that also in this case, collagen 1, 6, and 12 are the most abundant ones and produce much more abundantly compared to other CAF subsets with different functions. So now, why is that important that collagens are so produced so abundantly? Well, this is because collagens have a very particular amino acidic composition. So while they form this very complex structure, they are actually quite simple in terms of amino acidic sequence because they have tripeptides that are very frequently repeated. And among these tripeptides, we have one that is composed by glycine, proline, and hydroxyproline. What are the consequences of this is that if you compare the frequency of the different amino acids in collagen proteins here in violet compared to the frequency of the amino acids in the rest of the proteome, you can see that glycine and proline are much more abundant in collagens. So really, this raises the questions, can we target the availability of specific amino acids to disrupt collagen production? And this is what I will show you today. So this story started from a phosphoproteomic comparison of normal fibroblasts and CAFs. And we collaborated with the Julio Saez Rodriguez lab, and they use mathematical modeling to predict the kinases highly deregulated in CAFs upon activation. And what these studies found is that the pyruvate dehydrogenase kinase 2 was predicted to be the most downregulated inactivated kinase in CAFs. Now, the pyruvate dehydrogenase kinases are kinases that are inhibitory for the pyruvate dehydrogenase complex. This complex is very important because it basically transforms pyruvate into acetyl-CoA in the mitochondria. Acetyl-CoA then can be used in the TCA cycle, or it can exit the mitochondria, and then it can be transformed back into acetyl-CoA, and this acetyl-CoA can be used either for protein acetylation or for the synthesis of other molecules, including fatty acids, sterols, and ketone bodies. So what we wanted to test first was whether this pyruvate dehydrogenase was indeed more active in cancer-associated fibroblasts. To do that, we use an enzymatic assay, and what you can see here is that indeed the calves have the pyruvate dehydrogenase that is more active compared to the normal fibroblast. So what I told you before is that this enzyme converts pyruvate into acetyl-CoA, and we wanted to see indeed if we could see this metabolically. In order to do that, we did the tracing experiments where we feed our cells with the carbon-13 labeled glucose. And what happens here is that you can trace the fate of this glucose in this way. So you see that now it goes through, is used to make all the different molecules of the glycolytic pathway, and then it goes into pyruvate and acetyl-CoA. So basically, if we have an increase in activity in pyruvate dehydrogenase, we should see more glucose converted into acetyl-CoA in our calves. And this is what you can see here. So basically, when we did this experiment, we found that the calves have overall more acetyl-CoA. And if you focus on the color part, most of it is produced by from glucose via pyruvate dehydrogenase activity. So, but what we also know is that there are more pyruvate dehydrogenase kinases. So the model predicted pyruvate dehydrogenase kinase 2 to be more active, but there are actually 4. So we looked at the expression of all these different kinases in our normal fibroblasts in calves in different patient samples. And what you can see here is that what we found is that the pyruvate dehydrogenase kinase 2 is typically the most highly expressed in fibroblasts, and that all the pyruvate dehydrogenase kinases are expressed at very low levels in calves, at least in our modeling culture. But then using laser capture microdissected data from triple negative breast cancer, and this is a laser capture microdissected stroma of normal breast or triple negative breast cancer breast, we confirmed that indeed in tumors, pyruvate dehydrogenase kinases are very low expressed in the tumor stroma, and that compared to normal, the stroma of normal breast, PTK2 is highly downregulated. So basically now we have a model that actually recapitulates what probably goes on also in patients. So highlighting the relevance of our discovery. So, but can we then modulate the metabolism of the calves simply changing the levels of the pyruvate dehydrogenase kinase 2? So to answer this question, what we did was to overexpress PTK2 in our calves, and this was sufficient to increase the phosphorylation of the pyruvate dehydrogenase, that I remind you is inhibitory, and this indeed induced a decrease in production of acetyl-CoA. Conversely, if we take our normal fibroblast, we silence PTK2 to reduce its level, so we mimic what happens in the calves when they are activated. This is sufficient to induce an increase in acetyl-CoA production. So now the question is, where is this acetyl-CoA going? I told you before, it can be used for protein acetylation or for other metabolic reactions. So, and what we found, very interestingly, performing a global protein acetylation analysis of normal fibroblasts and calves, is that the calves have histones that are more acetylated. And here I highlighted a subset of these lysines on histone H3 that are known to be acetylated specifically by the histone acetyltransferase EP300. So this was a very exciting result for us because there is already literature in cancer cells and other models showing that an increase in cytosolic acetyl-CoA can result in increased histone acetylation, particularly HS3 that I showed you before, and also an increase in activity in the histone acetyltransferase EP300. But what is more important is that there is additional literature, particularly focused on fibrosis, showing that histone HS3 acetylation and the activity of EP300 are upstream of an increase in expression of profibrotic genes, including collagens. So we wanted to see whether this was true also in our system. So what we did here was a proteomic analysis of our CAFs upon inhibition or not of the histone acetyltransferase P300. And what was clear is that the majority of the collagens were highly downregulated. And you can also see this here in this immunostaining, where in blue you see the nuclei of our CAFs, and in red you can see the collagen. And what is very important here is that we could also demonstrate that this decrease in collagen production is due to a decrease in mRNA level, so in its expression. But something that we found very intriguing and very novel is that in addition to find the collagen highly downregulated, we'd also found this protein PYCR1. Now, PYCR1 is an enzyme involved in protein synthesis. It in particular is a mitochondrial enzyme that catalyzes the last step of protein synthesis, particularly from glutamine. So this was very interesting for us because, as I told you before, we know that collagens contain a high abundance of proline amino acids. So we wanted to understand whether there was a link between collagen production and this increase in potentially proline synthesis. So, and the first evidence that there might be a connection between these two mechanisms came from these experiments in which we took our normal fibroblasts. We silenced PDK2 that I showed you before induces this increase in histone acetylation and collagen production. Here you can see that indeed we can induce by simply silencing PDK2 increase collagen production. But now if on top of PDK2 we silence PYCR1, so we reduce the availability potentially of proline, you see that there is a decrease in collagen production. So this is the first link that there might be a connection between these two mechanisms. And this is depicted here. So just to summarize here, what I showed you so far is that in CAF activated, we have an increasing acetyl production because of deregulation of PDK2. And this triggers an epigenetic regulation that increases expression of collagens and protein synthesis enzyme. Now, is there a connection between these two? So what happens? Now, we were very interested in PYCR1 because it is one of the metabolic enzymes known to be deregulated, upregulated in most tumor types. What this plot shows you is that now if we look at gene expression data that have been previously published, we can see that both in the epithelial compartment and stromal compartment of breast tumors, we can see that there is an increase in PYCR1 expression both in the epithelial and in the stromal compartment with the increasing aggressiveness of breast cancer. And we can see that there is also an increase in collagen production in the stroma. So again, a nice link between protein synthesis and collagen. And importantly, what we found is that if we separate the patients that have invasive ductal carcinoma, we divide those with high PYCR1 and collagen 1 expression from those with low PYCR1 and collagen 1 expression. Those that express at high level both genes are those that actually have a poorer overall survival. So, and this for us is interesting because it might highlight that there could be actually a subset of patients that could benefit of an anti-PYCR1 therapy when we target both the tumor compartment and stromal compartment. So we now wanted to see whether indeed the CAFs were making more proline, so if the enzyme was working. So to do that, we did tracing experiments. So as I told you before, proline comes from glutamine. So we can now give, here on the right, we give a heavy labeled glutamine, and then we can trace heavy labeled carbons in a way that we can basically see whether the glutamine is used to make a proline. So what you can see here is that doing this experiment, we could indeed prove that the CAFs have overall more proline, but that also that they produce more from glutamine compared to the normal fibroblasts. And we could also show now that if we inhibit PYCR1, we can significantly decrease the production of proline from glutamine, either if we silence PYCR1 or if we use an inhibitor for PYCR1. But now the point is we wanted to see whether this proline was actually used to make a collagen. So to do that, what we did was to grow our cuffs in culture in the presence of heavy labeled glutamine that can be then transformed in heavy labeled protein that we know. So we cultured the fibroblasts in a way that they could produce over a few days extracellular matrix. Then we decellularize the extracellular matrix and analyze it by mass spectroproteomics. And then what we looked was at the presence of heavy labeled proline in collagen-derived peptides. And actually, this is what we see here. So basically, this shows us that the calves are using available glutamine to make a protein that is then used to make collagen proteins. But we wanted to see whether this was true also in an in vivo situation. So what we did here was to transplant our mice with human breast cancer cells. And then when the tumor was grown, we infused these mice with heavy labeled glutamine. And then we analyzed the blood in the tumor by mass spec metabolomics. So just to see that the glutamine was indeed in the circulation and in the tumor, and also to show that it was converted into proline. And this is what is shown here on the top. But what was more important is that we also analyzed by mass spec proteomics these tumors. And what we looked at was at the presence of heavy labeled proline in collagen proteins. And this is actually what this plot shows. So basically, this is the first proof of principle that mice can actually use, the tumors actually can use circulating glutamine to make a protein that is then used to make collagen. So, but what happens then to the collagen when we inhibit PYCR1? Now we take a step back again and we go back in vitro to our cups. And what is shown here is that when we inhibit PYCR1, we can actually significantly decrease the production of collagen. So follow the red staining that is for collagen 1, and you can see that indeed is decreased when we inhibit PYCR1. But what is important is that we can restore the production of collagen when we add exogenously supraphysiological levels of proline. So what is important here is that only adding supraphysiological levels of proline, we can actually restore proline, collagen synthesis, suggesting that the cuffs really require their own proline to be able to support the production of tumor collagen. And we know that this is dependent upon, so that this defect in collagen production depends on the inability of the cuff to translate the mRNA into protein. In fact, we didn't see any difference in mRNA levels when we inhibit PYCR1 in our cuff. So here is collagen 1 as an example. However, what this plot shows is that we found that the ribosomes actually stalled, particularly when there were protein codon, and that among the most affected proteins, we have a collagen. So we really have a significant reduction in the translation of collagen mRNAs when we inhibit PYCR1, and we can rescue in part this effect by giving the cells exogenous proline. What happens now in an in vivo situation? Now, what we did here was to basically transplant our mice with the patient-derived cuffs, either control or silence for PYCR1, and we could transplant that with the human breast cancer cells. So then what we looked at was at the presence of collagen. In this case, we look at fibrillar collagen by second harmonic generation and at the presence of cuffs. And what we found is that also in vivo, we could see a significant decrease in fibrillar collagen in tumors in which we transplanted cuffs silenced for PYCR1, while the amount of cuffs didn't seem to change. And here you can see a nice microscopy images of collagens and cuffs. But what happens to the tumor? So what we found is that when we inhibit PYCR1 in the cuffs, we found a significant decrease in tumor weight and tumor size. But we also wanted to see whether these had any effect in the aggressiveness of the tumors. So for this, again, we co-transplanted our mice with the patient-derived CAFs and aggressive metastatic 41 breast cancer cells. Also in this case, what we found is that we found a decrease in collagen production when CAFs were silenced for PYCR1, no differences in CAFs. But what was striking here is that also we found a decrease in tumor size and tumor weight, but very importantly, we found a strong decrease in lung metastasis in both circulating cancer cells and cancer cells that could colonize the lung. While we didn't find any difference in blood vessels and hypoxia, suggesting that this effect is due to the inability of the cancer cells to intravasate and form distant metastasis. So just to summarize, what I showed you today is that we have found novel mechanisms through which CAFs can promote and support tumor aggressiveness. We have discovered that the metabolism is very important to support the production of collagen. We have identified increased production of acetyl-CoA to trigger an epigenetic regulation to increase the transcription of collagen molecules and protein synthesis enzyme. And I also show you that the increasing production of protein is essential to support the production of tumor-promoting collagens. And I also show you that targeting PYCR1, we can inhibit cancer cell growth and the formation of lung metastasis. So, and with this, I wanted to thank all the people that have worked at this project. I wanted particularly to highlight NDK, who is a postdoc in my lab and did most of the work that I showed you today. But I also wanted to thank all the other groups at the Whitson Institute and our collaborators in the UK and other international collaborators. And I also wanted to thank you very much for your attention, and I'll be happy to answer any question later on. Thank you very much.
Thank you very much, Dr. Sada. Our next speaker is Dr. Simi Ali. She is Professor of Immunobiology at the Institute of Translational and Clinical Research, Newcastle University, UK. Can you please start her talk?
Hi, so I'm Simi Ali. I'm Professor of Immunobiology. I'm based at Newcastle University, and I would like to thank for the opportunity to present my work at the Shaukat Khanum Breast Cancer Symposium. So today I'll share our data on the contribution of glycosaminoglycan binding in CCL21 mediated migration of breast cancer cells. And I hope you will find the data interesting. So to give you a brief background, in cancer, the lymph node metastasis consists of a key event in breast cancer progression. And chemokines, they are small chemotactic proteins, and they can promote the metastatic spread by inducing cancer cell migration and invasion. The chemokine function is dependent not only by binding to its 7-transmembrane spanning G protein coupled receptor, but also to heparan sulfate molecules, which are negatively charged molecules found on the cell surface of endothelial cells. So our group has demonstrated a significant increase in chemokine receptor CCR7 expression in cancerous breast epithelial cells compared to the healthy controls. So this shows how we think the CCR7 has a role. CCR7 and CCL21 have a role in breast cancer metastasis. So what you see here is a blood vessel. It's lined by endothelial cells. And if you think of a primary tumor, so we have observed that there's increase in expression of CCR7. And its ligand is CCL21, which is expressed in the lymph nodes. So the cancer cells usually behave in a manner analogous to the leukocytes. So these cells can then migrate across the endothelial cells into the blood vessels and then extravasate into the tissue, into the lymph nodes, which express high levels of CCL21. So because CCL21 are involved in the lymph node metastasis, so our hypothesis was that if we can block this interaction, then we can reduce the metastasis. And I'll talk about our strategy of how we want to block this interaction. So to give you a bit more background on the interaction of CCR7 and CCL21 in cancer. So CCL21 and CCR7 have been shown to promote growth and metastasis in many tumor types, including breast. CCR7 is an important marker in the prediction of auxiliary lymph node metastasis in breast carcinomas. And overexpression of CCR7 correlates with larger primary tumors and deeper lymphatic invasion and poor survival rates. There are in vivo studies where they have shown that if you knock out CCL21 expression, there is decrease in metastasis. Similarly, CCL21, CCR7 plays an important role in the lymphangiogenesis, which is associated with pancreatic cancer. And it is involved in the lymphatic spread of the melanoma cells. So again, to give you a brief background about chemokines, so they are abbreviated from chemoattractant cytokines. There are about 50 different human chemokines which have been identified so far. And they are small molecules and they are structurally related. And this is shown here in a very simple diagram. And I'll just focus on the first two, the CC and CXC, because most of the chemokines belong to these families. And the way they are classified is on the presence of these cysteine molecules. So if the cysteines are adjacent to each other, they are known as CC chemokines. And what it means, and these chemokines usually, members of this family, usually attract eosinophils, T cells, B cells. And CXC chemokines, here there's an intervening amino acid between the cysteine residues. And the members of this family predominantly act on neutrophils. In addition, the chemokines are also divided functionally into homeostatic and inflammatory chemokines. And as the name suggests, homeostasis is it's a migration of naive T cells, B cells, and inflammatory chemokines are usually upregulated in response to an infection. They play a fundamental role in host defense mechanisms, and they're also important in the development, homeostasis, and function of the immune system. So I've just shown you that the subfamilies are divided on the basis of the presence of the cysteines. And as I showed you in the previous diagram, the CXC chemokines act on neutrophils, the CC act on the eosinophils. There are two other families, but they have very few members. And for the purpose of this talk, I'll not go into that. So how do these chemokines mediate their effects? So they mediate their effect through binding through 7 transmembrane spanning G protein coupled receptors. And as it's shown here, the chemokine interacts with the N-terminal domain. Then the G proteins interact with the C-terminal domain. The receptors have variable expression, and there is a range of complexity and diversity. So in addition to binding to the 7 transmembrane spanning G protein coupled receptors, chemokines also interact with molecules known as glycosaminoglycans. And these are carbohydrate molecules. They are negatively charged. They can potentiate the chemokine activity. And again, if we can block this interaction, it will stop the chemokine interaction with the cell surface of the endothelial cells and stop the gradient formation. So just to show you what are glycosaminoglycans. So this is a section of a blood vessel. And In the magnified form, you can see it is lined by endothelial cells. And these endothelial cells on the cell surface have proteins, and to them are bound the carbohydrate molecules. And these carbohydrate molecules are negatively charged. There can be several kinds of carbohydrate molecules, but the most abundant are heparan sulfate, which are found on the endothelial cells. So I'll use them as an example when I show you the data. So the aim of the work I'm presenting is to test the hypothesis that if you generate a chemokine, CCL21, which does not bind heparan sulfate, can this disrupt the response to normal CCL21, which is expressed in the lymph nodes? And this should then therefore reduce the metastasis of CCR7 expressing breast cancer cells. So first, we looked at the expression of CCR7 in human breast cancer tissue. We looked in several cancer types like ductal carcinoma, lobular carcinoma. And what it showed was that during the cancer formation, there is upregulation of CCR7, and there is variable expression, both in terms of the level of expression and the location. So it can be expressed intracellularly or on the cell surface. So then we generated a mutant CCL21, which cannot bind heparan sulfate. And for this, what we did was, this is the amino acid sequence of the CCL21. And what we're seeing in red is the region which has got a lot of positively charged amino acids, by which it binds to the negatively charged glycosaminoglycans on the cell surface of endothelial cells. So we created a truncated version where we deleted the C-terminal end. So once you make a mutant CCL21, the first question you have to ask is it biologically active? Can it still bind to its receptor? Can it still bind to its glycosaminoglycans? So the first experiment we did was to look at the calcium flux. And what it means is when functional chemokine binds its receptors which can be on leukocytes, or in this case, we've used a human breast cancer epithelial cell line. Then can it signal through the cell? And if it signals, there is increase in calcium. And what you see here is a representative figure which shows that when you add wild type CCL21, there is an increase in calcium following it binding to its receptor. And this is the tabulated version. And you see here that the wild type CCL21 can induce significant calcium flux at both the concentration, whereas the mutant shows much reduced at the lower concentration. So then we went on to see that we are creating a mutant which should not interact with the heparan sulfate or the negatively charged molecules on the endothelial cells. So does this mutant then not interact? And to do this, what we do is we use an assay which is known as an SPR assay, or surface plasmon resonance. And in this, we have chips, microfluidic chips, on which we attach the heparan sulfate. So what you see here is when you flow the wild type CCL21 through these chips which bind heparan sulfate, they have a significant level of binding. So this shows the KD on and off. Whereas with the mutant CCL21, I mean, you can see the axis is here, it's like 10-fold or 20-fold less. There is much significantly reduced interaction with heparan sulfate. So what this shows is the mutant we have generated is biologically active in binding to its receptor, but it is not able to interact with the glycosaminoglycans. So then we tested the mutant in the migration assays. So these are the assays which all the chemokine labs would do to ensure the biological activity. So this is just to show you what the assay involves. And it's a very simplistic diagram. It just shows you. So what it involves is you have got a well. In that you put an insert, and in this insert at the bottom end you have holes, and for purpose of breast cancer cells, the holes would be about 8 micrometer, so that the 10 micrometer cell can squeeze past it. The breast epithelial cells are added on the top. The chemokine is added at the bottom. The whole thing is incubated in an incubator for 2 hours. Then the cells at the bottom, which have migrated in response to the bottom, are quantified. And what it shows here is that when we use this system, which is transfilter, with PBNC with the mutant CCL21, there is reduced migration at 20 nanomolar concentration. With the MDA231, there's also reduced migration at 20 nanomolar. Then we wanted to do a migration assay, which is more physiologically relevant. So in order to do that, what we do is we line the filter with, we grow endothelial cells on the top. So it's basically like your blood vessels. So to go for metastasis, a cancer cell has to pass through the blood vessel and go into the tissue. And then when we did this here, we found that at both the concentrations, the mutant CCL21 supported significantly reduced cell migration compared to the wild type. So far I showed you that we made a mutant CCL21, and it has shown us in vitro assays that it does not bind glycosaminoglycans. It binds the receptor, but it shows much reduced migration and calcium flux. So next we wanted to test it in an in vivo model of breast cancer metastasis. So for this, we needed a cell line which expresses chemokine receptor CCR7. So for this, we screened a range of cell types available. And what we found is this murine cell line, which is murine epithelial cell line 41, expresses significantly high level of CCR7 at protein level, which was assessed by flow cytometry and by PCR. Then we also further characterize these cells, because once the cells are going through a cancer formation, the epithelial cells can undergo mesenchymal transition. So what you see here is these 41 cells express the epithelial markers, ecogerin and ZO1, but they also express some mesenchymal marker, which is vimentin, and they express human CCR7. So with this data, what we derived was that we have a cell line which expresses the CCR7 and it expresses the receptors. So it is probably suited for testing in vivo. So we also use the cell line to see that how does it respond in migration, and similar to what the data I showed you earlier with the human breast epithelial cell lines, the murine breast epithelial cells also responded significantly less to mutant CCL21 compared to the wild type. And this is just to show you that what does the filter look like, when the cells have migrated. So this is by background migration you have. This is migration in response to mutant CCL21, and this is a migration response to wild type CCL21. So you can see that the migration response to mutant CCL21 is much reduced compared to the wild type. So the 41 cells we also tested them using a more physiologically relevant model of transendothelial migration. And when we do this, what we do is we label the cells with a tracker dye so that you can differentiate the endothelial cells, which they are passing through to go to the bottom of the filter. And again, what we saw was that the mutant CCL21 supported much reduced migration, as seen here, compared to the wild type. So then these cells were transfected with luciferase because we wanted to assess the growth of tumor in vivo. And for that, we use IBIS system. And this is just to show you that the level of luminescence is directly correlated with the cell number. So that gives you the confidence that if you can measure how much the luminescence is in the tumor, that would correspond to you can extrapolate the number of cells in the tumor. So this shows the animal model we used. We used biopsy animals, and they were injected with the 41 cells in their mammary fat pad. And after day 7, they were treated with mutant CCL21, 20 micrograms IV, and they were treated every day for 7 days. And then on day 18, they were sacrificed. The luminescence was measured and the lymph nodes extracted. So that just shows you that how we monitor the tumor growth with IBIS. And we used, we carried out initial experiments with range of cell numbers for 41s, ranging from 50,000 to 2 million. And we monitored the growth with the luminescence every week for 3 weeks. And what we derive from here is that 50,000 is the optimum number, because at day 7, the tumor becomes palpable and the animals still last till day 21. Whereas with higher numbers, we had to sacrifice animals earlier because the tumor grew more than 1 centimeter in either direction, at which point we had to humanely kill the animals. So this shows you that how we identify the metastasis by IBIS system. So the animal is, when the skin is taken out, you can see this is the primary lymph node. And these are the lymph nodes, are the draining lymph nodes where the tumor is metastasizing to. And then we looked at the luminescence in these lymph nodes. And on basis of this, we derived that the lymph node 1 and 2 were the ones which were the draining lymph nodes, and they will be assessed for our final experiment. We also looked at the survival curves after injection of the 41 cells. And again, we found that 50,000 cells were the optimum for in vivo monitoring of the tumor. So when we administered mutant CCL21 and we isolated these lymph nodes at day 18, what we found was that the animals which were given the mutant CCL21 compared to PBS had a significantly reduced luciferase expression. And also, when you normalized it to GAPDH, so we have looked at all the lymph nodes together or just the draining lymph nodes. And in both the cases, we found that the level of expression was low. So what it shows was that the mutant CCL21 blocks the interaction with wild type CCL21, and it reduced the metastasis formation. So in order to summarize the results I've shown that CCR7 expression is increased in cancerous breast epithelial cells compared to the healthy cells. I've also shown you some data on breast cancer epithelial cells, MDA, MB231 migrate in response to CCL21. And then in terms of the mutant CCL21, I showed that it is biologically active. It induces transfilter chemotaxis, but significantly reduced trans-epithelial chemotaxis. And there's an additional work which I've not shown is some mechanism that how it causes the receptor internalization, more internalization compared to the wild type. So to conclude, what I presented today is that the mutant CCL21 is significantly decreases lymph node metastasis of 41 cells compared with PBS control. And this was after daily injection IV for 7 days. And it also shows that there's a potential for mutant CCL21 to be used in therapy, and particularly in combination with other drugs like checkpoint inhibitors. So I thought this diagram probably proposes the mode of action of mutant CCL21. And what it shows is that it's representing a blood vessel lined with endothelial cells. The red shown here is the wild type CCL21, which binds to the glycosaminoglycans or heparan sulfate, which is negatively charged, and it can then bind to the receptor on the cancer cells. And these cells can then migrate into the tissue, and then they can get to the lymph nodes and proliferate and form metastasis. Whereas if you give mutant CCL21 as a competitor, because it doesn't bind to the heparan sulfate, it can only bind to the receptor. It causes the receptor internalization, but the cancer cell cannot follow the gradient and cannot enter to the tissue into the lymph node to cause metastasis. So this is how probably it reduces the lymph node metastasis. So I just put this slide up to show the implications of the study. There are several studies which have shown that how wild-type CCL21 can be used in cancer immunotherapy as a chemoattractant for dendritic cells and T cells. There's a phase 1 clinical trial where they have done intratumoral injection of CCL21 gene, modified dendritic cells in lung cancer cells, and they have shown specific immune response and CD8 T cell infiltration. And there's also anti-tumor response in patients receiving checkpoint blockade may be further improved when combined by either in situ vaccination of CCL21 or probably combined with the mutant CCL21 we have found. So finally, I'd like to acknowledge everybody who's been associated with this group, my longtime collaborator, Professor John Kirby, Dr. Annette Neeson, and Irene, who did most of the work, and also the funding bodies. And thank you for your attention. And thank you again for giving me an opportunity to share my data.
Thank you very much, Professor Simi. Our last speaker is Dr. Farhana Badr. She is Senior Biostatistician and Cancer Epidemiologist at the Department of Cancer Registry and Clinical Data Management in the Shaukat Khanum Memorial Cancer Hospital. Dr. Farhana.
Assalamu alaikum, ladies and gentlemen. I have been asked to give a brief presentation on the topic of cancer registration. As most of you are aware, we have different types of cancer registries. We have hospital-based registries, we have population-based registries, and disease-specific registries. Here I'm just talking about the population-based cancer registration sponsored by the Shaukat Khanum Memorial Trust. We have actually two. One is the Punjab Cancer Registry, and the other is the Northwest Cancer Registry. The Punjab Cancer Registry was established in the year 2005. It has over 40 members in about 27 collaborating centers of Lahore. Most of them are in Lahore, but there are some that are also located in other districts of the province as well. And these include both the hospitals and labs, representing the private sector and the government sector. So what we do is to use both the active and passive form of data collection. And we also have a mobile data application, data collection app. So that has been very helpful as well. And Children's Hospital Lahore has been doing a lot. They have been sharing the data using this application. And eventually the data is entered into the software within the hospital information system. So we give monetary incentives to data collectors of government hospitals as well, because logistically it has been very difficult to convince staff of other hospitals and labs to do this. Although we have been working for over 15 years, we have managed to collect data for Lahore, and I think we have done a good job, but we need to focus on other districts as well. So at present, our coverage is 19 of 36 districts. Punjab has 36 districts, and at present we are covering nearly 40% of the population. And this is how it goes. We're attempting to make it a contiguous cancer registry in terms of data collection as well. So you can see that we have Lahore over here, and the population density is very high in Lahore. And you can see that the areas of the other districts are high, as compared to Lahore, and the population density is low over here. But we yet have to get all the information related to this and prepare reports. Although we have presented one on Sheikhupura that is available on the Punjab Cancer Registry website as well. And at present, we are working on Faisalabad and Nankana Sahib. So here we have Lahore. We already have worked on Sheikhupura to some extent, and then we are currently working on Nankana Sahib and Faisalabad. And we are hoping to work on other districts in due course of time. So this is just meant to give you an idea of how it has been from the beginning. In the year 2005, the collaborating centers barely contributed over 2,000 cases. And over the years, it has been going up, as you can see. And in 2021, nearly 7,000 cases were reported. This is 2022, and we are still getting cases for this year, but we have already collected information on over 5,000 cases representing the district of Lahore, and this is exclusively for the district of Lahore. Now about the other districts of Punjab, and this information is for the year 2021. You can see that Faisalabad has contributed a lot, over 2,000 cases, but for the rest of the districts, it's less than 1,000 in each one of them. Although Sialkot and Gujranwala have contributed over 700, but for others, it seems to be going down. So we would request our collaborators to please share the data with us, so that we can have information related to these districts as well, and we can get the incidence rates and move on from there to program planning and evaluation using the data provided to us. So, and the report has already been published. This is a 10-year report. It's been published in BMJ Open. So the second registry, I'll go through it quickly as well, is the Northwest Cancer Registry. And we have included in it Khyber Pakhtunkhwa, FATA, which is now a part of Khyber Pakhtunkhwa. We have Gilgit-Baltistan and over here, and the Azad Kashmir region as well. So these 4 regions are included in the Northwest Cancer Registry. So it was set up in 2015, much later than the Punjab Cancer Registry. And in terms of the cancers reported, you can see how it has gone up. From 1,400, it went up to 4,500, then 7,000, then 8,000, and it's around 7,000-8,000. It has been so for a few years, but I'm quite sure that the cases are still being underreported, and we have requested centers representing the Khyber Pakhtunkhwa province to please report the cases to us, and they have assured us that they are going to do that during this year or the next year, even for the years 2021. And the Pakistan Atomic Energy Commission has also been on board, and they have also showed their willingness to collaborate with us, although in the past it was not so. So hopefully these figures will also improve. So what are the different ways in which one can report the cases to the registry? Registry officers visit different hospitals and labs and distribute the data collection forms and collect them where needed as well. So these forms are also emailed to the data collectors. They guide the collaborators about data capture in the fields that are required to be filled in. The required fields are name, age, phone, address, and the clinical diagnosis. And these fields are also required to check for duplication within the registry. And at times, data collectors also take photos of the filled out forms and send them to us electronically, and at other times in the form of an Excel sheet. They fill it out and then they send it to us. So as I just said, we have a data collection application as well, and all of these methods are used both for the Northwest Cancer Registry and the Punjab Cancer Registry. And eventually data are entered into the software within the hospital information system. So we are giving monetary incentives, and we have developed a structure for that. It depends on the number of forms they fill out and share with us. And these are meant for representatives of the government hospitals. And doctors and members who contribute significantly to the registry are included either as co-authors or in the acknowledgement section of the articles that are written on cancer registration. And this is just a repetition what I said. We need this data for program planning and evaluation, the numbers of hospitals needed within the country, consultants, nurses, etc. There is a dearth of this data and it is badly needed. And I'm hoping you will pass on this information to your friends and colleagues so that they can also share the data with us. This is the contact information, so we can move on from there, and cancer registration must go on. I also have the data collection form over here if somebody is interested or wants to share it with their colleagues, both for the Punjab Cancer Registry and the Northwest Cancer Registry. So that is all I have for today. Thank you very much for your attention towards the end of the session.
Thank you, Dr. Farhana. Thank you. I would like to express my sincere gratitude to all the speakers for their excellent talks. Now we are going to start the question answer sessions. Kindly raise your hand if you have any question.
So I have two questions, one from Dr. Farhana. That's excellent registry presentation. So you just mentioned that you are sending data registry officers to different hospitals. So like we have now a very well-established COVID interface. So everything which is being reported anywhere in the hospital, it's automatically going to the central system. So maybe what do you think about establishing something similar? So any patient diagnosed with cancer should automatically, that information should be shared with Punjab or KPK registry?
But they haven't yet done it. So it depends on when they're motivated enough to do it. But it's a very good idea. Yes, it goes there directly.
My question is also for Dr. Farhana. Excellent presentation and such great work for cancer registration in Pakistan. So like for most countries with high cancer burden, national cancer programs are suggested, and it is considered that it's crucial planning to implement those programs. So see that one of the limitations other than underreporting and other challenges that you face is the availability of human resource for cancer registration. Would you suggest what can Pakistan do to develop our human resource capital for cancer registration, our capacity building for that?
I have written that in my publications as well. At the district levels, we should have well-trained data collectors. We should have medical coders there. And they should know how to do the data entry in the software that has been developed. And from there, it can come into the centralized registry, as Dr. Ispan said. And then from there, it can be taken forward to the National Cancer Registry that they are doing it. But it has to happen at the district level. They have to take charge of it. Otherwise, it's really, really difficult for the data, for the cancer registry officers. They spend the entire day going to different hospitals and labs as well. And at times the data collectors are not available. I mean, there are different scenarios because we have been doing that for 15 years now. So it's a lot of effort. Yes, it should be like that at the district level. We should have trained staff for that purpose.
Excuse me. I have a question. Do you have any mechanism to avoid the multiple entries of a data? Like, for example, if a patient is registered somewhere in Jhelum and then he comes to a lab like Chuktai and then he comes to Shaukat Khanum, then is he entered like 3 times, or is there any like mechanism to avoid these multiple entries?
check for duplicate records or duplication, we use a combination of variables. We could have name and the cancer type. We could have age and the cancer type. So we use a combination of variables to see if the patient has been entered more than once. In the past, we wanted to have the CNIC, but that hasn't happened. Because many centers say that it is difficult to get the CNIC number of the patients. So we've stopped doing that. It was a required field actually initially, but it is no longer the case.
Yeah, because without CNIC number, if you are taking other variables, they can overlap in many patients, like multiple times.
But we do check for duplicate records, and at times it is 15 to 20% of the total cases reported. And that used to be with the Pakistan Atomic Energy Commission data. Inmol used to report a lot, but they stopped doing that 3 to 4 years ago. But now we have convinced them and they are on board now. They are sharing their data. So that is the only mechanism we have, because we don't, at times we don't even have the phone numbers. So we either return the forms and ask them to get the phone numbers, because that is also one way of checking for duplicate records. We could have name and phone number. And we do use a combination of variables, and that is written in our publications as well.
Okay, thank you.
So I have a question from Professor Yasik Gronwald. A very informative research work you have presented. Just a comment, have you noted the differential pattern of micronutrients with disease recurrence or distant metastasis, number one? And secondly, like for other biomarkers, clinicians are checking on every chemotherapy cycles and based on that tailoring the therapy. So what about checking micronutrients level on every cycle or after every year or so, just to decide how long a clinician should offer a treatment? Thank you.
Thank you for question. Yeah, so we make quite recently, it is not published yet, extensive study on ovarian cancers. And it is big, and blood sample was taken during diagnosis. And it is big association between staging and so selenium level is very low in 3rd and 4th clinical stage. It is statistically higher if the clinical stage is 1 and 2. So in ovarian cancer, we observe it. Yeah. And anyway, looking at the mechanism, yes, so selenium is incorporated into selenoproteins, and so it looks that the cancer is metabolizing. Cancer cells are metabolizing the selenoproteins very quickly, and that's why the selenium level is dropping down. Yeah, this question, yes, it is a very good question about chemotherapy and after every cycle, what happens, yes. It should be tested, so we don't have very big experience about, so regular measurements. It is, so from one extent, if cancer cells are damaged, so, all these microelements can be spread everywhere, and it must be measured, and some associations must be done, and then it may be some clinical impact on these measurements.
I have a question. Excuse me. In your initial slides, you have shown that with the high levels of zinc and selenium, they were associated with a high risk, high lifespan. But have you considered, like, those values were high, like really high, because in humans it's like 100 mg per microgram per liter of selenium, I guess. But you showed 2000, something like 1000 or 900 something, yeah.
So in selenium, it is over 100. It is, it depends on population. For example, Poland is the country with a relatively low level of selenium, let's say medium level of selenium, and it is about over 70. It is medium, but in the United States, it is over 140. What
Okay,
with zinc,
Mhm.
so it is, several thousand.
but
yeah,
with these high level of zinc and selenium, have you checked that if there were any side effects or any risk factors or tissue toxicity which is related to the high dose of these
No, in
micronutrients?
general, we don't observe any side effects, yes, because, it was not supplementation. It was, these measurements were done in consecutive cases of patient cases. So
Mhm.
when they were coming with, let's say, breast cancer and so on, so it was the measurement. And it is sometimes a little bit higher or lower than, let's say, medium.
Okay.
Yes, so for selenium, so it is, we expect, and it's that around 100 for women with, healthy women with this from family with breast cancer, so it is optimum. So
Okay.
if in respect of cancer, for example, there are some patients with some thyroid problems. And it is suggested to be a little bit higher, or let's say 140 and so on. So we have to take into consideration what is the major problem of a patient. Yeah, but looking in, let's say, population point, yeah, so it should be, let's say, 100. If it is in family, the largest problem of breast cancer, yeah, so BRCA carrier, so familial familial breast cancer. So this around 100, I think it should be recommended. Yeah.
Okay, thank you.
I have a question for Dr. Sara. Is she here? As you said that collagen is involved in tumor microenvironment and can influence the cell behavior. So do you think that like now these collagen induction therapy and supplements and using in beauty products can impact on occurrence of cancer in female? And can these products avoided by these cancer survivor patients? Do you think?
Sorry, I could not hear the entire question. Could you repeat?
Yes, I can repeat, sure. As nowadays, these induction therapy, collagen induction therapies, treatment supplements are very popular nowadays.
Oh, I see.
Treatment. Do you think that these are avoided by the cancer survivor or patients or female, like it can involve in tumor microenvironment? So do you think that should be avoided?
So I think that's a very interesting, I haven't thought about it, but as far as I'm aware of, there is no evidence, right, that taking collagen can have an impact on tumors. So I don't even know if, the collagen that you are taking in that way is actually going potentially in a tumor, or how much it's changing the microenvironment. I think it's a good question, but unfortunately I don't have an answer for you.
Thank you.
Okay, thank you. And with this question, we will conclude this session. Thank you very much for all our speakers, both online and in person. And also to the audience for their active participation.
Thank you. Thank you very much.
Thank you so
Thank you
much.
very much.